imaging session. My name is Qi Wen. I'm associate professor in biomedical engineering department. As you probably know, brain is the most complicated organ in our body. Okay, so Columbia has very strong neuroscience and neuroengineering program. And today we have a great lineup of speakers from Columbia School of Engineering, Columbia Medical School, and also Columbia School of Arts Sciences and the Zuckerman Institute. So they are going to tell us their exciting work about their how to understand brain. So our first speaker is Grace McVeigh. Okay. So she's a new assistant professor of our department. She's going to tell us brain biomechanics.
All right, thank you so much. Thank you for having me here today. I'm delighted to be kicking off our Engineering and Medicine Symposium. My name is Dr. Grace McElvain. I'm a brand new assistant professor here at Columbia University in the departments of Biomedical Engineering and Radiology. And today I'll be talking about my research in brain magnetic resonance elastography, which is a new and notable neuroimaging contrast. And I am looking to recruit for my lab, and I'm looking to form some new collaborations here at Columbia. So if you're interested in this work, please come talk to me. So one of our most valuable and really underrated tools in medicine is our sense of touch. By you or your doctor feeling lumps and bumps on your body, you can really get a pretty good idea about the underlying cellularity. So by measuring these mechanical properties, you can get a lot of information that you wouldn't have otherwise. But problematically, one of our most important organs, the brain, we really can't touch it. We can't figure out, what is the mechanical properties of a brain tumor. So my research is in doing exactly that. I develop non-invasive methods for measuring mechanical properties of the brain, one of them being magnetic resonance elastography. And MRE is a MRI modality in which we can create these whole brain maps of mechanical properties, including things like stiffness, damping ratio, viscosity, mechanical anisotropy, even poroelasticity. And how this works is we put somebody in an MRI machine. We gently vibrate the back of their head with this special pillow. Feels kind of like a cell phone vibrating on the back of your head. And from this, we create these micron level displacements in brain tissue. Why this works is because waves change their characteristics based on the underlying material properties. So you can see in this gelatinous phantom here, the center is more stiff, the outside is more soft, though both of these are within the realm of physiologically normal for the brain. And we can see even just by eye that the wavelengths are quite different in this more stiff area and more soft area. You can also see that you can really define even by eye where the margins of that inclusion are. So you can imagine with some advanced modeling software, we can really get a pretty good picture of how stiff or soft your brain is. And in the brain, we're doing this. We're measuring things like the storage and the loss modulus of the brain, and we create these whole brain maps of mechanical properties. Now, why we care about mechanical properties in the brain, what it tells us about the underlying cellularity is a bunch of things. So mechanical properties kind of describe this aggregate of both the mechanical properties of individual cells as well as how they interact to form a glial matrix. So we believe mechanical properties relate to things like the number of neurons that you have, with more neurons being a good thing and making your brain more stiff, the amount of myelin that you have, which is a fatty coating on the outside of an axon, again, making your brain more stiff, and then the density of the glial matrix. But if you are somebody who is well-versed in MRI, you're probably thinking, well, we can kind of measure these things via other MRI methods. We can measure the contrast, kind of gives us a good idea of density, things like that. But why mechanical properties are special and important is because they can tell us this unique aspect of tissue that really no other measure can. So it's this somewhat arbitrary concept of like, how well are all the neurons hanging on to one another in the brain? So in this image, there's actually the same number of cell bodies in both images, but you can imagine that they have very different mechanical properties. The one on the left has these nice connections between cells, while the one on the right is, much lower integrity. And why this matters is because if you're thinking about neurodegeneration or even neurodevelopment, cells don't just appear out of nowhere or die instantaneously. They start to lose their interconnectedness before they'll die. So we think that mechanical properties are really sensitive to these early stage changes, and we can measure it with elastography. And we can see this. We see that brain decreases in stiffness with healthy aging. It's just inevitable. Your brain's getting softer. We also know that different neurodegenerative diseases, Alzheimer's disease, multiple sclerosis, Parkinson's disease, these things all accelerate the rate of stiffness decline in the brain. Very interestingly, we also see that mechanical properties are related to the function of the brain. So we see these really defined structure-function relationships. Here you see that the mechanical properties of the hippocampus are related to how good somebody is at memory. And this is in people ages 18 to 30 years old. So healthy young people, we see these structure-function relationships in the brain. And we see these oftentimes despite lack of changes with volumetric measures, which volume is a very standard neuroscience tool that we use to look at degradation, but we can see mechanical property changes even when volume's not changing. So we think mechanical properties are really sensitive to these structure-function relationships. Brain MRE is a really new modality. So if you haven't heard of it, no problem. It started in just about 2010, but it was this low resolution, single slice, took a long time to scan kind of modality. And it wasn't very useful for the clinic. So it's really been in the last 10 years or so that we've developed these tools that are now able to produce fast acquisition, high resolution, whole brain property maps that are useful for clinical things. Though while we've made a lot of progress, there's still a lot of work to be done. So about half of what my lab's research is in the technical development of brain MRE, making this tool better. So can we correct for subject motion? Can we improve SNR? Can we make it higher resolution, faster acquisition, things like that? And then we take all these technical advancements that we work on, and we use it to answer this big question about how do the mechanical properties of the brain reflect underlying brain function and brain health? So can we non-invasively measure these mechanical properties and describe the underlying cellularity? Because of course it's impractical to take biopsies and look at cellularity directly. So this is a non-invasive method. We want to understand if mechanical properties are reflecting these things we care about. And one of the examples of this is looking at people with multiple sclerosis. So we know in chronic MS that the brain stiffness is reduced, but about 80% of people with MS have this relapsing remitting form of MS. It's kind of like the precursor to full stage MS. And during this, people have these big demyelinating events, followed by periods of remyelination. And every time the brain remyelinates, the myelin comes back weaker and weaker. And so this is kind of what the disease trajectory looks like. The problem is in the remyelination phase, it's really hard on conventional MRI to describe that loss of integrity of myelin over time. And so we think that brain elastography can help us to understand, is someone's brain remyelinating 99% of the way or 50% of the way? And what is that telling us about their specific disease trajectory? Another area I do a lot of work in is brain tumors. And we know that brain tumors have different mechanical properties. Meningiomas are stiffer than normal tissue. Gliomas are softer than normal tissue. I've done some work in low-grade gliomas where we see even in these, stage 1 and 2 brain tumors, we can see the differences in mechanical properties. You can see the brain tumor is very soft. Again, here's another example that the glioma is very soft. Finally, an example in the cerebellum. We're seeing these soft brain tumors, even though they're these low-grade gliomas. We've also seen that tumors can have very high heterogeneity in terms of their mechanical properties, which is important because if you're a neurosurgeon trying to remove this or trying to understand, is this, one cell type in the tumor, or are there different components, you might want to understand this difference in mechanical properties. And why mechanical properties are different in gliomas is because of the underlying cell type. So healthy tissue has these nice, strong connections between cells where glioblastoma cells are the large, phylomorphic kind of cells. The cell people are going to be mad, but they're kind of just like stacks of water, right? They don't have this nice, strong structural cohesion. And so with this, we hope that we can do things like measure, is the tumor infiltrating? Can we see changes in mechanical properties from these infiltrating cells? Can we measure inflammation and necrosis, which happen a lot of times because of radiation treatment? Do these processes change tissue mechanical properties? And finally, one area that I do a lot of research in is studying healthy development. So the developmental trajectory of the brain is really important to understand healthy brain development and a whole host of neurological disorders. Pediatric brain MRE is brand new. So in 2018, we wrote the very first paper. There have been a handful of papers since then. I actually forgot to put the most recent papers on here, but the field is really growing at looking at how does the brain develop mechanically, and what does this tell us about structure-function relationships in the brain? So we've seen that in healthy people, executive functioning measured by the Stroop and the flanker task is related to the mechanical properties of the thalamus. We have seen that in children ages 5 to 7 years old, the Broca's area or the right inferior frontal gyrus is related to how good somebody is at vocabulary. We've also seen that in adolescence, the difference in mechanical properties between your risk-seeking and reward suppression regions of the brain correspond to mechanical properties. And so why these relationships are important to understand is because we can see a lot of these things through fMRI. But fMRI is not a quantitative technique, and it's also not good for people who can't engage in tasks. So, you can't put a 3-year-old in an MRI scanner and expect them to play vocabulary games while they're in there. So having these measures of tissue structure is really important for accomplishing a lot of our goals in understanding healthy development. So in summary, brain mechanical properties are really important for understanding the brain. Brain elastography is this up-and-coming technology that's really great. And I am recruiting for the lab. So if you have any interest in studying the mechanical properties of the brain, please let me know. All right, thank you.
Thank you. Okay, our last but not least speaker is Professor Olson from Department of Auditory and also Department of Biomedical Engineering.
Great. Oops. Okay. So before getting into that, I just wanted to tell you a little bit about how the healthy ear operates. So you can see over there the ear, the outer ear, the ear canal, and when a sound pressure wave comes into your ear, it causes the eardrum, which is at the very end of the ear canal, to vibrate with the sound, and that causes those little middle ear bones, which are in an air-filled space on the other side of the eardrum, I think you can see, to vibrate. And finally, the third one that is going into the bone depicted there in blue, which is the inner ear, vibrates and causes pressures within the fluid of the inner ear. And the snail-shaped part is the auditory part of the inner ear, and here is a cross-section of that. And so it's the white part is fluid. You can see the sensory tissue in the middle of the fluid, and there's something pointing to it there in pink. And so the stapes would plunge in and out of the cochlea here at the base. That would cause vibration within that, the sensory within the fluid, which is in sync with that pressure, but it's separated by the cochlea into different frequencies. So the high frequencies cause vibration mainly in the base, and then low frequencies go all the way to the apex, the tip up there. And and the auditory nerve spirals up, or goes up the middle, and then the auditory neurons project out to contact the sensory tissue. Okay, so here's an image of the sensory tissue, just a flat one, and then the one on the left gives you an idea of the sensory tissue going back into space, going spiraling along and along. And at the top of the red and the blue are the hair cells, which are the transducer cells of the cochlea. They're the things that turn mechanical motion into an electrical response. And that happens via the stereocilia, which are the hair at the tip of the hair cells you can see on the right. And they're actin rods that tip back and forth in sync with their vibration. And you can see they're joined to adjacent stereocilia are joined by a tip link, which opens up like a little trap door. So we have current going through the hair cells. As those trap doors open, close, they cause current to vary through the hair cells, which causes voltage variations through the hair cells, and that leads to neurotransmitter release from the bottom of the hair cells to the neurons, and then action potentials, neural spikes in the auditory neurons. Just a short detour here to regarding otofurlin gene therapy. You might have heard on the news that there was gene therapy that actually cured deafness recently, and that was the otofurlin protein is something that is at the synapse there that lets the neurotransmitter go out from the hair cell to the neuron. So it's a very specific deficit, and miraculously, it was actually like these kids couldn't hear and then they can hear. So that was something that happened the past months and was, ongoing, exciting research. Most people who are deaf, the problem is in more of the tissue, in the hair cells might be missing or very damaged. And for those folks, gene therapy is still something for the future, but for now, they would get a cochlear implant. And a cochlear implant is you can see how it looks here on these two, a kid and maybe her dad or a man. And so a cochlear implant bypasses all of that mechanical stuff that I just told you about. And instead, it picks up the sound sort of like a hearing aid would. You can see the microphone is labeled there. It's on the part that's sort of looping over his external ear. And so the sound is picked up by a microphone. It's processed by the speech processor, like a little computer. And then there's a battery there too. And then there's a transmitter to transmit it into the internal part, which is the receiver stimulator, which is connected to this electrode array, which has been threaded into the cochlea. So you can see those dots going into the spiral of the cochlea. Each dot is a different electrode. The sound, just like in the natural ear, is separated into different frequency bands, and the higher frequencies are at the base, closer to the stapes. The lower frequencies go to the apex. And this is the most common neural implant. More than 1 million have been implanted worldwide. It's by far the most successful neural implant. So what this talk is actually about is making an implantable microphone as a component of a fully internal cochlear implant. This is our team from Columbia, MIT, Harvard, and Mass Eye and Ear. And the benefits of a fully internal cochlear implant are many. In general, it's convenient. You don't have to, there's no handling required, changing of the battery, et cetera. There's 24/7 here, and you don't remove it for sleeping or swimming and things like that. So it's good, safer, and just easier. Of course, there's a cosmetic benefit if you don't like the idea of having this thing on your head. Specific to an implanted microphone, there are also other benefits. There are acoustic benefits of if you can put the microphone within the middle ear space or the cochlea, then you'll get acoustic benefits from the external ear and the ear canal, which provides some pressure amplification and also gives you cues about where the sound source is. It also eliminates problems that external microphones have due to wind noise and head coverings. So the main challenges to a fully implanted cochlear implant are two. One is you need to wirelessly charge it because the battery has to be inside. And this is actually an area of rapid development. We're not doing that. But for many implants, that's something that needs to be done, and it's being done. Number two, we need to figure out how to put the microphone into the body. And this is what our team is working on. And fully implanted cochlear implants are already in advanced development. They're already in clinical trials. Medell says the future of cochlear implants is invisible cochlear implants, meaning fully implanted. So where should we detect the sound signal? Where should we put our microphone? And the options are subdermal, like underneath the skin, within the middle ear space, and within the cochlea. And our microphone detects a sound signal in the middle ear, at the middle ear bone that is attached to the eardrum, which is called the umbo. So the actual displacements of the umbo are sub-nanometer to hundreds of nanometers. So very small motions that we are going to be detecting there. So how should we detect the sound signal? We need to convert this umbo motion into a voltage. And the way that we're doing that is, this is an image of our patented and current, design, which we've been testing now for a couple of years. And it's based on a PVDF, which is a piezoelectric polymer, and made into 2 layers so that we can do a differential amplification in order to reduce some of the noise. And it's tested, here's a picture of it in, placed into the middle ear space. The ear canal would be above. This is the umbo, which is the middle ear side of the eardrum, and you can see those other sort of sculpted things are the other ossicles going to the cochlea. And the testing of the microphone output is done in human temporal bones, which are donated for research. And so here's just one data slide. This is on my right, your left, is an image of just data from 5 sensors just to show how repeatable the results are. I can't remember what the pressure was, but it was sort of a normal sound pressure. And so the human up on the top is an audiogram, and the important thing there is just to see that conversational speech is from around 100 to 10,000 hertz, so that's the frequency range we really need our microphone to work well. Humans hear up to 20,000 hertz, a little higher than that, especially if you're young. So you can see that there's good repeatability, and the drop off above 4 kilohertz is really due to the umbo motion, not due to the microphone itself. But it's adequate up to 10 kilohertz. On the right is equivalent input noise, which is a common way that hearing aid microphones are evaluated, which is what we need to compare to. So in the light blue is our microphone. In the black is a Knowles microphone from a hearing aid. So you can see ours has a little bit higher equivalent input noise, which is a measure of noise over signal. But if we include, here in the testing, we've gotten rid of the pinna and much of the ear canal. So if we include their effects in our device, then the dark blue curve would result, which would mean that our noise is at the same level as this microphone. And I'm going to try to play this. In addition to doing this more objective measuring, we also just plain old talk to the temporal bones and record the signal, and then I think...
Hello, I'm about a meter away, and I'm talking at a normal conversational level.
Okay, so that's a recording that we made with this microphone, just to give you an idea for the sound quality. So the next steps are a lot of hairy details, making the thing fully bicompatible, encapsulating it fully, really figuring out how to fix it to the middle ear space, making sure it would last for 50 years, and a large animal study, and also trying to make the amplifier lower power. And I'll just end with something that we've been mainly working at here at Columbia, which is developing sheep as a large animal model for microphone implantation. The reason that you'd want to do a large animal model is because one of the big problems with, this is my last slide, by the way, one of the big problems with an implantable microphone is body noise. And when you do a test in a temporal bone, you're really not getting that. So by implanting in a large animal, you'll get an idea for like how much chewing and blood flow and breathing and stuff would impact in a bad way your microphone. So Nick Waring is in the back there. He was a medical student who really did an amazing job getting sheep heads and just figuring out how to get the temporal bones out, and then working on the surgical access. And then the fixation device has been worked on a lot by Xiaoshen Zhao, who's a mechanical engineering graduate student. And so that's it. Thank you for listening.
Okay, so can we ask all the speakers come to front? We are going to open to the floor for questions from audience. We're going to have a 10 minutes panel discussion.
And we also have one virtual question.
Okay.
We want to start with that?
Yeah, maybe we can start.
Okay, great. I believe this is for Grace McElvain. Are these mechanical changes able to detect cell growth as in adult neurogenesis? What is the resolution currently?
So the resolution, the best we can do is about 1.25 millimeter resolution. We have not yet done things like specifically trying to grow more cells and measure changes to mechanical properties. Everything that we've done is anecdotal in saying, we see in Alzheimer's disease decreases in brain stiffness, and we know in Alzheimer's disease you see cell death. So I would say it's kind of the other way around. We haven't done those prospective studies yet.
Hi, can you hear me? I was just curious about any collaborations within Columbia you have, and Just talking more about that, if you could. Just
For me?
everyone.
Oh. Well, I can start by saying I just started here in January, so don't have many collaborations yet, but I'm working on forming collaborations, and so far Columbia has been really wonderful in that the faculty have reached out to me quite a lot and said, lots of ongoing work, so I'm excited about the opportunities for collaboration.
I collaborate with somebody in electrical engineering and also in mechanical engineering and otolaryngologists. There are a lot of, as you heard early on by the deans, that they were not lying. There's a lot of collaboration, so it's very easy to collaborate across campuses. And
yes. Brain surgeons, brain tumor people, people who are looking at development of fish hearts, lots and lots of people. I mean, our lab, has attracted a lot of people, obviously from neuroscience, who want to sort of use our techniques. But I would say that the best collaborations have kind of actually come from where really groups work together, like students actually kind of pair up and work one from one lab and one from another. And so some of our best collaborations have been, that kind of really authentic collaboration and not just sort of people coming to use our stuff, which is very helpful, but really, really good when we make it work. And I collaborate with people as well across the city as well, like Mount Sinai, things like that. There's loads of hospitals in New York that have different levels of expertise in different things, and we are able to collaborate with them also.
Yeah, I feel the same. I think it's Columbia is a really great place, especially biomedical engineering department as well, to kind of being exposed to a lot of different domains. So we collaborate a lot with pathologists to get whatever brain samples, postmodern brain samples, or collaborate with geneticists or for stem cell, iPSC lines, and so on. Right. So it's really broad expertise in this area, which you have access to, depending on what you need.
Yes, we have a question online for Elizabeth Hillman. Can you please elaborate to the applicability of your technologies to autopsy human tissue and what you would hope to study?
So those human brains come from autopsy, obviously, but actually, so I'm really, we had a paper last year, I think, where we showed that our fast microscope actually works inside of a living human. So we imaged like the tongue and showed that we could actually visualize that in 3 dimensions. And so what I really want to do is start to do histology like in the living body, like during surgery, and not have to cut the tissue out and have it die and then go off and try and image it, right? And if we do the imaging in vivo, we can actually pick up information about tissue function, perfusion. And so that's actually something we're pushing and we want to actually develop commercially. So that technology is also great for what we call bedside histology, which is sort of very rapid screening of tissues that have been cut out, if that's where we have to go with it. And so, yes, it could be also used at autopsy. But yeah, so we do that too.
Okay, we probably have time for the last question. Anyone from audience?
Thank you. This is a question for Dr. McIlvaine. From what I understand from your talk, you say that the mechanical properties of the brain and the interconnectedness can be indicative of executive functioning. So I was wondering if that means that with this technology we can look into autism, for example, or ADHD.
Yes, so autism is something that I have been excited about looking at for a really long time. I think there's a lot of opportunities, not actually even in diagnosing autism, so I don't claim to be able to do that, but I think autism is a highly heterogeneous disorder, as you probably know. Some people have major deficits in language, some people in behavior, and I think it's probably possible to measure the mechanical properties of the brain in a young child with autism and say, okay, this child has deficits in their Broca's area. They are somebody who might struggle with language as they get older. Let's hurry up and give them language intervention now to try to, remediate those facts. So autism is definitely a big application of this work.
Great. Let's give our speakers a round of applause.